All right, now we're going to focus in on mitosis. So our book breaks mitosis down into 5 phases. If you've seen mitosis before, you might have seen PMAT, which is prophase, metaphase, anaphase, telophase. We have kind of broken prophase into prophase and then prometaphase, which is kind of early prophase and then later prophase. Some texts you might call this early prophase, and then prometaphase might be called late prophase, and then they skip to metaphase, anaphase, telophase. So there's just kind of 2 different conventions for naming the different phases here. All right, so here we have the G2 of interphase. So this is gap 2 of interphase. This means we have not started mitosis yet. This is still interphase. The cell is doing its normal thing. It's growing. It's doing its normal processes. We have, in animal cells, we have centrioles. These occur in pairs, and they're kind of here at 90 degrees. And these are going to help organize the spindle fibers. The chromosomes are uncondensed. So in here, you can see all of these lines. They look just like skinny spaghetti. The nucleolus is still there. The nuclear envelope is intact. Plasma membrane's out here. So the cell looks pretty normal. It's not doing anything weird yet. It's not dividing. Now we're going to go ahead and start with mitosis, with prophase. So in early prophase, we're going to start to see the spindle fibers kind of separate. And these are going to eventually move to kind of a north and south pole on the cell, but they're not quite there yet. The spindles are going to form between these. So what actually happens is these spindles contact each other and they push against each other. And that's actually what moves the centromeres around. The spindle fibers are going to be our movers. Those are what's going to grab onto our chromosomes and actually move them around. Notice the nucleus is starting to look different. The nucleolus is gone, so there's no nucleolus here. The nuclear membrane is still intact. So the nuclear membrane is still intact. We're not going to be able to move those chromosomes around yet. The chromosomes have started to condense. So over here they were uncondensed, and now they're starting to condense. You can see distinct lines. So if we go ahead and count these, I see 1, 2, 3, 4, 5, 6 centromeres with duplicated chromosomes here. They're still pretty long. They're not quite condensed. This one's really long. They're going to get more dense as time goes on. The reason the chromosomes have to condense is because they have to move around in the cell. Imagine trying to separate all of these thin pieces of spaghetti. It's like if you've ever sorted necklaces and they're just all tangled up together. What you really want to do is condense them all and then organize them. So think about when you move from one house to another house. You don't just throw all your stuff in the car. You condense it down into boxes first, and then you move the boxes. So that's what's happening. We're condensing the chromosomes down into movable sizes. So the next phase here is prometaphase. Sometimes you'll see this just called late prophase. We can see that those centrosomes have moved to the opposite poles. So now we have kind of a north and a south, and that's going to tell us we're going to divide this way and this way. It kind of gives direction to which ways the cell's going to divide. These are highly condensed, so these are much, much smaller, and these are now kind of in movable little packets. They are connected to the spindle fibers. So go back to our moving analogy. If these are the boxes that you're moving, then the spindle fibers are the movers. The spindle fibers are what are going to actually pick up your boxes and move them to your new house. And you can't leave your house unless you open the door and leave. So in this case, the nucleus, right, so our nuclear envelope is going to break down and disappear. We've got to get rid of the nucleus completely so that we can reorganize the genetic material, and then we'll make some new nuclei later. Next we have metaphase. Metaphase, I like to remember the middle. Metaphase has 2 characteristic processes that are occurring. The first here is that all of those chromosomes are lined up on the metaphase plate. And they've kind of drawn this as a line here, but it's more of like a 2-dimensional plane where all of the chromosomes are lined up in the center of the cell. And the reason they're lined up in the center of the cell is because the spindle fibers from each side are connected to the kinetochore proteins. And the kinetochore proteins are just on the sides here that are what going to connect to the spindle fibers. And the reason they line up in the middle is because they are connected on both sides. And it's kind of like tug of war. So the spindle fibers are pulling equally on both sides of the chromosomes, and that's going to allow it to kind of end up in the center of the cell. The next phase is anaphase, and ana means up from. I always associate anaphase with movement. This is where the chromosomes are going to physically break apart. So you can see that we've broken these apart here. They are not connected on that metaphase plate anymore. They're still connected to the spindles. The spindles are actually going to shorten and start to pull these apart away from the center. So if we count the chromosomes now, we can count how many centromeres we see. And we've got 6 on this side and another 6 on this side. So we went from having 6 duplicated chromosomes to now having 12 single copy chromosomes. And those are going to move towards the 2 poles of the cell. And you can see the cell is actually starting to elongate. Some of these spindle fibers are just pushing against each other, and that's going to help make the cell longer. The last phase of mitosis is telophase. The word telo means end, like the end of a telephone. When telephones used to have cords that went into the wall and you talked on the end of the cord. Telophase is only concerned with what's happening in the nucleus. The nucleolus is going to start to reform. The nuclear membrane is going to start to reform. Look at these chromosomes. They're starting to get longer and longer. So they're going to start to uncondense. The spindle fibers are gone. So those yellow spindle fibers, these guys, they're gone. We don't need the movers anymore. Once we've moved into 2 new houses, we don't need the movers. They don't just hang out. They go away. And so we've got our centrioles for our new cells. Cytokinesis is a separate process from mitosis. It usually does start to occur during telophase. So we've got cytokinesis here. In an animal cell, what's going to happen is there's going to be a ring of filaments that comes around the center of the cell here, and they're going to get smaller and smaller and smaller until eventually the plasma membrane kind of pinches off, and we end up with 2 separate cells. So the centrosome is found in animal cells. Plants still have spindle fibers, but they don't actually have the centrosome that organizes them. And what this does is it creates those microtubules, all of these spindle fibers. And these were the movers that are moving the chromosomes around the cell. I like this image because you can really see the metaphase plate here, that it's really just a 2-dimensional plane through the center of the cell. And it's not like a straight line. They're not necessarily organized in a straight line here in metaphase. A kinetochore is a protein complex that's assigned with a centromere. So if we've got the centromere here in the center, the kinetochore is the proteins right there on the side, and then that's where the spindle fibers are going to go ahead and come in and attach, and that's how they actually move the chromosomes around in the cell. This is just like the movers grabbing onto the handles of a box and then moving the box around when you move houses. During anaphase, when the chromosomes separate, they are cleaving proteins called cohesins. If you think back to water coherence, right? Coherent was water sticking to itself. So cohesin is kind of like a glue. These are proteins that hold the 2 chromosomes together. So these cohesins are going to be right here in the center. So when those are broken, you're going to end up with 2 chromosomes that are completely separate because we broke the cohesins that are keeping them together. And then the way that they get pulled apart is that the motor proteins that are holding on to the kinetochore actually walk the chromosomes along the microtubules, and then the microtubules depolymerize. So I like to think about this like, if you've ever played a video game like Mario, where when Mario jumps on the platform, he's able to land on it, but the second he jumps off of it, the platform falls down. As soon as the motor protein has moved past a point, the polymers that used to be here break away and depolymerize. So they're just going to break off monomers. And we know that the polymers actually shorten at this end rather than this end through some experiments. During this experiment, what they did is they actually marked kind of the center of the spindle fibers here. And then throughout anaphase, as the chromosomes moved closer and closer to the ends of the cells, we were able to see that the polymers over here were being removed, whereas over here it had stayed the same. So they're actually depolymerizing at the end over here and not shortening at the end over here. After mitosis is over, we're going to go into cytokinesis. And so you saw cytokinesis kind of starts usually during telophase. And animals and plants are going to have two very different methods of doing cytokinesis. Animals do not have a cell wall. So animals, all we have on the outside of the cell is the plasma membrane. So we form a contractile ring of microfilaments here. And that ring just gets smaller and smaller and smaller. And then eventually it's going to pinch these two cells into two to separate those membranes. Plants, on the other hand, have a cell wall. Right? So a cell wall is a very sturdy structure on the outside of the plasma membrane, and it's going to help plant cells keep their shape. It helps keep them from bursting, and it's very strong. So that cell wall is not going to be something we can just kind of pinch into two. So instead of forming a cleavage furrow, the plants actually have to form a cell plate, which is a brand new cell wall on the inside between the two cells. So it starts as little vesicles. You can see these little vesicles here, which are going to be the basis of the new cell wall. Those vesicles get larger and then eventually merge into a cell plate. And then that cell plate will eventually merge with the existing cell wall to create a continuous wall around each of the new daughter cells. We talked specifically about mitosis being in animals and plants. Prokaryotes also have the capability to divide from one cell into two identical clones, and they do so through this process called binary fission. Bacteria do not have linear DNA, they have circular DNA. And on that DNA, they have a specific region called the origin of replication. So what ends up happening is they need to copy their circular DNA. They get two copies of it, and then they're able to divide into two cells. The two molecules of DNA are going to move apart from each other. We believe that the origin of replication helps in organizing this. When the two circular DNA molecules move apart, what we end up seeing is that the origin of replication ends up towards the poles of the cell. That's west and east poles. And then eventually the plasma membrane is going to kind of pinch inward, very much like a cleavage furrow. And then eventually we end up with two bacterial cells with copies of the original circular DNA. Binary fission is a little bit simpler than mitosis. It doesn't have quite the complexity of steps. It doesn't have the spindle fibers. So we think that binary fission probably evolved before eukaryotes, which makes sense because prokaryotes evolved before eukaryotes. And then mitosis probably evolved from binary fission as a method of dividing cells that are eukaryotic and more complicated.